Hello, fellow warriors. Here we are with news from the website prostatewarriors.com. Welcome to this custom deep dive designed specifically for you.
Absolutely. It's great to be going through this with you all.
Yeah. Imagine fighting an enemy that, magically changes its armor every single time you switch your weapon. That is essentially what we face with hormone resistance in prostate cancer. You block the fuel and the tumor just figures out a new way to survive.
Right. It's incredibly adaptive.
Exactly. But today, we're looking at the new weapons designed to actually shatter that armor. We've got a stack of crucial updates from the absolute front lines of research. Our mission today is to really break down how this battlefield of treatments is fundamentally evolving.
And it really is a turning point for the science right now.
It is. And we are following a very strict roadmap today, basically mandated by the science itself. We're going to cover the clinical trials first, starting with the massive phase 3 studies, and then we'll work our way down to early phase 1 trials.
Yeah. And then from there, we'll transition into some, honestly, truly futuristic preclinical science happening in the lab right now.
Right. It's a lot of ground to cover, but it's all about equipping you with a grounded, hopeful understanding of where this fight is actually heading.
I know the sheer volume of research can feel, completely overwhelming at times. But when you step back and look at the progression from the clinic to the lab, a very clear narrative emerges. We aren't just finding new ways to poison cancer anymore. We're actually figuring out how to outsmart it.
I love that. So let's head straight to the front lines. We're starting with a massive global phase 3 trial, and it's called Himalayas-1.
Right, the Himalayas-1 trial.
And when I say massive, I mean we're talking about approximately 600 patients over 250 sites spread across more than 20 countries. The goal here is to tackle that exact armor-changing problem I mentioned earlier, hormone therapy resistance.
Yeah, they're testing a drug called Rinzimetostat. You might also see it in the name RMAC944, and they're testing it in combination with Darolutamide.
Right, and just to clarify, this is specifically for men with metastatic castration-resistant prostate cancer, or mCRPC, who were previously treated with abiraterone.
Exactly. This is a highly strategic combination, and it gets right to the heart of how prostate cancer adapts. So darolutamide is a potent androgen receptor antagonist. Basically, it cuts off the signaling the cancer relies on to grow.
But, as we said, the cancer just changes its armor.
Right. When you block the androgen receptor, the cancer cells often activate, like alternatives, survival pathways. And that brings us to rinzimetostat. This drug blocks a protein complex called PRC2, specifically by targeting a subunit called ED.
Okay, wait, I've seen a different name in the research, too.
Yeah, you might have also heard of EZH2 in your reading. That's just another main component of this exact same PRC2 complex.
Okay, let's unpack this because I want to make sure I'm getting the mechanics right here. This PRC2 complex is essentially a tool the cancer uses to silence certain genes, right?
Yes, exactly.
Like specifically the genes that would normally tell the cell to just stop growing out of control. It's heavily linked to how tumors become resistant to hormone therapies. So, if darolutamide is cutting off the tumor's main power supply, the androgens is PRC2 sort of like the tumor's backup generator, kicking on to keep the lights on.
That's a great way to look at it.
Yeah.
What's fascinating here is the biology behind that backup generator. It's called epigenetic resistance.
Epigenetic.
Yeah, epigenetics is how cells control which genes are turned on or off without actually changing the underlying DNA code itself. So prostate cancer cells use the PRC2 complex to epigenetically rewire themselves so they can survive without androgens.
Oh, wow. So rinzimetostat is basically going in and cutting the wires to that backup generator while the darolutamide hits the main power.
Exactly. By combining darolutamide, which hits the primary pathway, with rinzimetostat, which disables that epigenetic rewiring, you're launching a simultaneous counterattack. You aren't just blocking the fuel, you're blocking the tumor's ability to adapt to the lack of fuel.
Which is brilliant. And what's really encouraging for the guys listening is the early data from the phase 1b dose optimization trial. The results were pretty striking.
They really were.
They saw a 59% PSA50 response rate, meaning 59% of patients saw their PSA drop by at least half. Even more impressive, there was a 24% confirmed PSA90 response rate. And crucially, the side effects like, gastrointestinal issues and fatigue were reported as highly manageable.
Yeah, and I imagine it's that balance of actual tumor control and quality of life that pushes the trial into a massive phase 3, right?
Precisely. The phase 1b data proved the biological concept was sound and, tolerable for the patient. Now, Himalayas-1 is designed to prove if this combination definitively extends progression-free and overall survival compared to our current standard choices.
If it succeeds, I mean, we're looking at a brand new oral strategy for the post-Aberrant setting.
Which is huge. Okay, so that's overcoming resistance with oral drugs. Now we're going to shift our focus to targeted radiation, specifically a phase 2 trial called Optimal-E.
Right, optimal.
This is a pilot study happening at St. Vincent's Hospital in Sydney, focusing on men with metastatic hormone-sensitive prostate cancer, or MHSPC. And we need to be very careful with the drug name being tested here.
Yes, definitely.
It's 177 ludodawinek pan-PSMA. You might also see it written as TLX597-TSM.
Right, the precision with that name is really important. So 177 ludoda pan-PSMA is a radioligand therapy.
Okay.
It's a small molecule drug designed to seek out and bind to PSMA, which is a protein found in, well, high amounts on the surface of most prostate cancer cells. When it finds the PSMA, it delivers a payload of lutetium-177.
Which is the radiation part.
Exactly. It emits beta radiation right into the tumor. The patients in this trial received this alongside standard androgen deprivation therapy and an androgen receptor pathway inhibitor.
But the part of the Optimal-E trial that really caught my eye isn't just the drug, it's how they're actually dosing it. They're using an adaptive dosing strategy guided by PSMA-PET imaging.
Right, the adaptive part is key.
So the patient gets the treatment and then they get scanned. If the scan shows the tumor is shrinking and the PSMA targets are reducing, they actually pause the treatment. They only resume the radiation if the patient's PSA starts to rise again and the PSMA targets reappear on a new scan.
That's the protocol, yeah.
But wait, I have to push back on this. Doesn't stopping the drug give the cancer a chance to, catch its breath? If you have the tumor on the ropes, why not just keep hitting it until it's entirely gone?
It's a very intuitive question, honestly. And it's how we used to think about cancer treatment universally. But it gets to the heart of how we have to manage radiation toxicity.
Ah, right.
You have to remember that while radioligand therapy is incredibly targeted, the radiation still circulates in the body. It clears through the kidneys, and it can affect the bone marrow and salivary glands.
So it's about minimizing that collateral damage.
Exactly. The goal of adaptive dosing is to maintain the patient in a low-volume metastatic state for as long as possible while significantly reducing their cumulative radiation exposure.
So for the guys listening right now who are worried about the long-term toll radiation takes on their bodies, I mean, this sounds like a massive quality of life upgrade. So it's really about playing the long game.
Yes, exactly. Think about it mechanically, right? If you just keep hitting a shrinking tumor with fixed, scheduled cycles of radiation, you might not be killing much more cancer because there are simply fewer PSMA targets left to bind to.
Oh, because the drug has nowhere to land.
Right. But you are still exposing the patient's healthy organs and bone marrow to that circulating toxicity. By pausing when the disease is controlled and only resuming when the targets reappear, you are maximizing the damage to the tumor while protecting the marrow.
That makes total sense.
Yeah, you're improving the therapeutic index, getting the maximum benefit for the minimum harm.
Work smarter, not harder. I love that. All right, let's dip down one more level in the clinical trial pipeline to phase 1. This is where a brand new mechanism of action is just entering human testing.
This is a really interesting one.
The trial is for a drug called SHYONC6, and it's an open-label, multi-center study for patients with advanced solid tumors, including MCRPC. This drug attacks something called the proteasome.
Right, the proteasome.
But before we get into what the proteasome is, your notes mention this is especially promising for PTN-deleted MCRPC. For those of us who aren't geneticists, what exactly is PTN, and why does its deletion matter here?
That's a crucial piece of the puzzle. PTNN is a tumor suppressor gene. You can think of it like the brakes on a car. It stops the cell from growing and dividing too quickly.
Okay, the brakes.
Right. In many advanced prostate cancers, those brakes are completely deleted or mutated. Because the cell is dividing so rapidly without any brakes, it's under massive metabolic stress. It's essentially running an engine way into the red zone, which produces a lot of damaged junk proteins.
Okay, so the cell is basically filling up with its own garbage because it's running too hot. That brings us to the proteasome. What does the proteasome do?
So the proteasome is the cellular machinery responsible for degrading and recycling all those damaged proteins. It's the cell's garbage disposal and recycling center.
Oh, I see where this is going.
Yeah, because PTN-deleted cancer cells are making so much junk, they rely heavily on this quality control system just to survive. If you block the proteasome, the cancer cell fills up with its own toxic waste and essentially triggers its own death.
Now, Older drugs targeting the proteasome, like bortezomib, have been around for a while, but they had pretty mixed results in prostate cancer, right? They didn't really move the needle.
Unfortunately, no, they didn't.
So how is SHY-ONC6 different? If the older drugs just jammed the main shredder in the recycling center, is this new drug tossing a wrench into a completely different gear in the machine?
That is exactly the hypothesis. The pathway that tags proteins for destruction, which is called the ubiquitin proteasome signaling pathway, is incredibly complex. It has multiple steps and different enzymes involved. The older inhibitors targeted very specific catalytic sites on the main proteasome barrel itself. SHY-ONC6 modulates the pathway through a completely distinct mechanism.
So it's a totally different approach to the same problem.
Right. By attacking a different vulnerability in that exact same recycling process, researchers believe it might overcome the resistance mechanisms that cause the older drugs to fail.
It's a great example of how a previously closed door in research can be kicked open again if you just forge a different key.
Perfectly said.
All right, we've covered the clinical trials, phase 3, phase 2, and phase 1. Now we are shifting our focus to the preclinical lab, where scientists are working on breakthroughs that will drive the trials of tomorrow. And the theme here is synergy.
Synergy is the perfect word for this.
We just discussed radioligand therapy, or RLT, in the clinic.
Yeah.
Now, let's look at how researchers are using RLT not just to destroy tumors, but to fundamentally alter them so the immune system can attack.
This is where the intersection of different scientific disciplines becomes really powerful. Prostate cancer has notoriously been an immune cold tumor.
Meaning the immune system just ignores it?
Basically, yeah. It means the patient's own immune system, particularly the T cells, either can't recognize the tumor or they are actively suppressed by the environment surrounding it, what we call the tumor microenvironment. Immunotherapies have historically struggled against prostate cancer for this very reason. The T cells just can't get in.
But researchers are finding ways to force their way in. In a recent preclinical study on MCRPC models, scientists combined radioligand therapy, specifically a 177-Lu PSMA-617, which many warriors know as Pluvicto, with a cutting-edge immunotherapy called CAR-RT cells.
Right, engineered CAR T cells.
And these specific CAR-T cells were engineered to target a different antigen called PSCA. They also tested this same combination strategy using a powerful alpha emitter, 225-ACU PSMA-617, and saw similar results. The combination drastically improved tumor control compared to using either the radiation or the CAR-T cells alone.
The results were really striking.
But here's where it gets really interesting, because this part genuinely confused me. The researchers found that the RLT is acting as an immune primer. Wait, if radiation is basically a destructive bomb, how does a bomb prime the immune system? That sounds totally contradictory.
It does sound contradictory until you look at how the cells actually die. If we connect this to the bigger picture, the radiation isn't just vaporizing the cells silently. It causes what we call immunogenic cell death.
Immunogenic cell death, okay.
Yeah, the dying cancer cells essentially burst open, violently releasing tumor antigens, inflammatory cytokines, and distress signals into the surrounding area.
Ah, okay, so it's less like a bomb and more like a flare gun. It's lighting up the area and sounding an alarm so the CRT cells can swarm in.
Exactly. That alarm bell activates the body's endogenous T cells and completely shifts that heavily suppressive tumor microenvironment. It goes from a state of immune exclusion, where immune cells are kept out, to a highly pro-inflammatory state.
So it completely changes the neighborhood.
It changes the neighborhood, right? It breaks down the chemical barriers that usually keep the immune system at bay. So when you introduce the engineered CAR T cells, after giving the RLT, they enter an environment that is finally primed and permissive.
And the fact that they're targeting PSMA with the radiation and PSCA with the CAR T cells, that's a dual target approach. So if a tumor cell mutates to hide its PSMA, the CAR T cells still catch it with the PSCA target.
Exactly. It helps solve the massive problem of heterogeneity. There's just nowhere for the cancer to hide.
Speaking of making sure the cancer has nowhere to hide, our final preclinical study takes an incredibly futuristic approach to that exact same problem of cold tumors. But instead of using radiation as a flare gun, researchers at Duke University are using CRISPR RNA editing to force the tumor to reveal itself to the immune system.
This study is an absolute masterclass in molecular biology. To understand this, we have to look at how prostate tumors become invisible in the first place. Normally, a healthy cell, and even an early cancer cell, displays proteins on its surface using a structure called MHC class 1. This is how the cell presents pieces of its internal proteins to the immune system.
Like a checkpoint.
Yeah, T cells constantly patrol the body, checking these MHC class 1 receptors. If they see a normal protein, they move on. If they see a mutated cancer protein, they attack.
Okay, so the MHC class 1 is essentially an ID badge that the cell has to show to the T cell security guards. But many prostate tumors figure out a way to lower their MHC class 1 levels. They just take off the ID badge and they slip right past surveillance.
Precisely. And the Duke researchers figured out exactly how the tumor throws away the ID badge.
Mhm.
It comes down to a protein called SPSB1.
SPSB1, okay.
In many prostate cancers, the messenger RNA, the mRNA that carries the instructions for making SPSB1, has an abnormally shortened tail. This tail is called the 3' untranslated region, or 3UTR. Normally, this tail contains control elements that tell the cell when to stop making the protein. But because the tail is cut short, those stop signals are completely missing. The cancer cell overproduces SPSB1.
And what does SPSB1 do?
It actively destroys MHC class 1.
Wow, that is a devious chain of events.
Yeah.
So the shortened RNA tail leads to way too much SPSB1, which then destroys the MHC class 1 ID badge, making the tumor invisible.
Yep, that's the mechanism.
So how do we fix it?
Hmm.
Well, the team at Duke developed a platform called 3UTRCS, and it uses a CRISPR tool, specifically a modified enzyme called DCAS13. Now, when most of us hear CRISPR, we think of Cas9, which acts like molecular scissors cutting DNA. But DCAS13 is different, right? It binds the RNA without cutting it.
That's the brilliance of this approach. It intercepts that shortened mRNA message and physically forces the cell to rebuild the full normal tail.
Rebuilds it, wow.
Yeah, by forcing the cell to rebuild the natural control elements on the mRNA tail, the production of SPSB1 plummets. When SPSB1 drops, the destruction of MHC class 1 stops.
Meaning the ID badges return to the surface of the tumor cell.
Exactly, and they prove this works in mice. When they package this CRISPR tool into lipid nanoparticles, very similar to how our mRNA vaccines are delivered, and combined it with standard immune checkpoint therapy, they saw remarkable results.
That's incredible.
Yet tumors that were previously completely resistant to the checkpoint drugs suddenly became sensitive. They turned a cold tumor hot, entirely at the RNA level.
That is basically hacking the cell's software. So let me make sure I've got this. The tumor takes off its ID badge to sneak past the T-cell security guards. This CRISPR tool doesn't alter the patient's permanent DNA hardware. It just intercepts the RNA software message and forces the cancer cell to put the ID badge back on.
That is exactly right. And that distinction between DNA hardware and RNA software is vital for the future of this technology.
Why is that?
Because the DCAS13 enzyme only targets the transient RNA messages and leaves the underlying DNA untouched. There is no permanent genome editing taking place.
So it's safer.
Right. It is theoretically much safer. It avoids the risk of off-target DNA mutations, and it's reversible. It represents a massive biotechnology trend toward editing the temporary messages of the cell rather than rewriting the permanent hard drive.
It's just incredible how these different approaches, from epigenetic backup generator drugs and adaptive radiation to immune priming and RNA software editing, are all attacking this disease from completely different innovative angles.
And that leads to the most important takeaway from all of these sources: the traditional silos of cancer treatment, where you just do hormone therapy until it fails, then you just do radiation, then you just do immunotherapy. Those silos are completely dissolving.
They really are.
We are entering an era where epigenetic drugs boost the efficacy of hormone therapies, where targeted radiation is used to physically prime the environment for CAR cells, and where CRISPR RNA editing is used to make checkpoint inhibitors actually work.
It's all connected now.
Exactly. The future isn't about finding one single magic bullet. It's about the brilliant orchestration of combinations.
That is such a powerful perspective. For you listening right now, the fellow warriors dealing with this day in and day out, consider how these synergistic, multi-layered strategies might completely redefine what it means to manage this disease in the near future. It's not just about blocking one pathway anymore. It's about outsmarting the cancer's ability to adapt, minimizing the toxicity you have to endure, and turning your own immune system into the ultimate weapon. We hope this deep dive has given you a clearer picture of the incredible, tireless science fighting on your behalf. Stay strong and fight on.